Hey everybody, it's Dr. Gilchrist, and as promised, I am talking about stereotaxic surgery and some other neuroscience methods today. So the last time that we were in class before we started playing with our fun brain toys is I was talking a little bit about the comparisons between MEG versus EEG. Now, as I kind of mentioned, both are looking at the same activity, but they're doing so in very different sorts of ways. They have the same temporal resolution because when you have those postsynaptic potentials, that creates a change in electricity, which will then simultaneously have those magnetic fields. So you're looking at the exact same thing in real time. So the temporal resolution is exactly the same because they are derived from the same activity. But because of the way that neurons are oriented in the cortex, as well as our good old right hand rule, where the magnetic field is basically perpendicular to the current, MEG often tends to be better for activity that is deeper in the cortex, like inside a sulcus. On the other hand, EEG is often going to be better for activity that is occurring right at the surface of the cortex in the gyri. So because of the way that our neurons are oriented, these are going to be used for different things and will be optimal for different things. Now, generally, we find that MEG, as I kind of mentioned last time, is pretty common in neurolinguistics research. And that's because both the inferior frontal sulcus, which is around Broca's area, as well as the superior temporal sulcus, which is near Wernicke's area, are both going to be very important for language. So now we're going to move on. And we're going to talk about transcranial magnetic stimulation. And I kind of talked about this a little bit. So we've talked about different measures of looking at real-time brain activity by looking at electrodes, either microelectrodes or macroelectrodes, as well as measuring magnetic fields. But some of the other ways that we can do biopsych research is by stimulating activity. And so generally, you can do this with transcranial magnetic stimulation. Now, usually transcranial magnetic stimulation, when it is applied, will impair an area, but it sometimes can facilitate activity in certain areas. So basically, what you're going to do is this little structure here is going to basically pass magnetic fields into the cortex. So you're going to take this thing that looks like a key, you're going to put it right on the surface of the scalp, and that is going to send stimulation through those magnetic fields. This happens basically by passing pulses of electricity through a wire coil. And remember that whenever we're passing electricity, we're going to get a perpendicular magnetic field. So as you can kind of see here, the electric current goes this way. The magnetic fields will be perpendicular to that and will affect the underlying brain area. And ideally, you would do this to interfere with the functioning of that area underneath. And the reason that you would want to do this is that it helps you learn about cause-effect relationships, or what role certain brain areas have for certain different types of activities. So this causes many neurons to fire if you're stimulating, or it can inhibit neurons if it is repetitive in nature. So oftentimes when you see transcranial magnetic stimulation being applied, you're going to get this clicking sound, which can actually be a little loud and a little bit annoying. And one of the other things that we'll find is that depending on where it's applied, it can produce muscle twitching. It can also, if applied to an area like Broca's area, although it would be on the left, wouldn't it? It's going to potentially produce a temporary speech arrest. So part of the reason that TMS is so cool is that it basically causes or produces temporary lesions, or under certain circumstances, it can facilitate activity based on how it is applied. So basically, we're asking ourselves this question: does disruption in area A impair behavior A? If it does, it probably means that area A is important for that behavior. Now, what's interesting, you may have heard about transcranial magnetic stimulation, particularly if it is repetitive in terms of being an actual treatment for depressive symptoms, as well as others like obsessive compulsive disorder. Generally, we're going to find that because this is repetitive, it can actually produce longer lasting changes, whereas your standard run-of-the-mill transcranial magnetic stimulation is basically those changes are only going to be present while that pulse is applied. We still don't quite understand how it works, but it's pretty experimental and we're learning more all the time. So we've talked about direct techniques. So we've talked about recording neural activity through things like EEG and MEG. And we've talked about enhancing or inhibiting brain activity with things like transcranial magnetic stimulation. Now, we are going to spend some time talking about ablation. So ablation is basically the oldest method in neuroscience that has been done to investigate the functions of the brain. And this has been used in both humans and in animals. Now, I want to be really clear. We do not intentionally ablate brain tissue in humans. We actually look at patients that have existing lesions or injured areas. So when you hear me talk about a brain lesion, we're talking about an area of the brain that is injured. So we're going to look at patients that have brain lesions to basically understand how the brain works when parts of it are damaged. So basically, one of the best ways to learn about what the brain is responsible for is to see what happens to those abilities when parts of the brain are damaged. So a lesion is an area of damaged tissue, and ablation studies are often going to be called lesion studies. So one major problem with lesion studies is that it's really tempting to basically think that this one brain region is critical for this process. The problem is that any one brain region does not act in isolation. All of the different areas of the brain are connected to other brain regions and through networks. So just because a brain area is damaged and a particular behavior is impaired, it doesn't necessarily imply that area is solely responsible for that behavior. It could be another connected brain region that supports the behavior, or it could be both of those regions taken together. So now we're going to talk about producing lesions in animal brains. So all of these are going to be accomplished by anesthetizing an animal, putting it into a stereotaxic device, which basically stabilizes the head, and it allows you to actually be precise when you are producing this brain damage. So we're going to start by talking about radiofrequency lesions, or RF lesions. These are produced by basically inserting a wire through the brain to the area that you want to ablate. You basically inject an electrical current into that wire, and the heat from that current will damage the tissues, including any neurons or glial cells that are nearby. So this is effective, but it's not very specific. It's not very efficient. It's often pretty crude because you're going to produce more damage than you actually want to have. So here is kind of an example of what this looks like. So here you can see the area where the wire was inserted, and you can see that the damage that has been produced through those radiofrequency lesions. So the next type of lesion that we are going to talk about is what is called an excitotoxic lesion. This is going to help you be more precise. So you're basically going to take a very narrow tube called a cannula. You're going to inject into that cannula an excitatory amino acid neurotransmitter. This neurotransmitter is going to bind to glutamate receptors. So it's going to produce EPSPs. The brain's going to get really excited, but these EPSPs are going to be firing at such a rate that it ends up destroying the cells. So one thing you need to know is that too much excitation for your brain is dangerous, and oftentimes it will kill cells because it's basically destroying the balance of the ions, it's damaging the ion channels and the receptors, and as a result, the cells are going to die. Now, this is more precise and more focal because it does not destroy all of the passing axons from other brain regions. It's just going to be the neurons in the area that you injected it into. Now, another method that's kind of like this allows the researcher to basically target different types of neurons that produce specific neurotransmitters. So for example, we're going to talk about 6-hydroxydopamine or 6-HD. 6-HD is a chemical that is similar in structure to both dopamine and norepinephrine. Remember that dopamine and norepinephrine are both catecholamines, and they have a very similar chemical structure. Norepinephrine is actually derived from dopamine. So the reuptake transporters for dopamine and norepinephrine, ultimately, when they see 6-hydroxydopamine, they're not going to be able to tell the difference between 6-HD and dopamine and norepinephrine. So 6-HD is going to bind to those receptors and those reuptake transporters. And then 6-HD is going to be taken back into the cell, and there it's going to produce free radicals, which damage the neurons. So this is just one example, but there are other types of drugs out there that are specific to different types of neurons. So for any type of ablation surgery that you're doing, it's really important to take into account that yes, you are damaging the brain, but you're also potentially producing damage in other ways. So think about what needs to be done when we're doing these lesions. You need to anesthetize the animal. You need to drill inside its skull. You need to insert the cannula. And let's be clear, any of these can produce damage even without the injection of current or of toxins. So if we want to assess the relationship between a brain region and its behavior, we need to try to control for these effects. So typically what you will see people who do animal research do is what is called a sham surgery. So basically what you're going to do is that you're going to have some groups of animals that get the lesion in the standard way, whether through radiofrequency current or excitotoxic lesions. But for another group of animals, what you do, these animals are going to go through the exact same surgical procedures, but without the toxin or the radiofrequency current. So they're going to be anesthetized. They're going to have drilling into the skull, and they're going to have insertion of a cannula, but they're not going to get the lesion. So then what you can do is you can compare your sham lesion group with your actual lesion group. And you can see if there are behavioral differences between the two. And if it turns out that your group that actually has the lesion looks differently from the group that has the sham lesion, you can basically conclude that it's not all of those additional surgical procedures that are producing those differences. So in order for ablation surgeries to be accurately performed, the head has to be put into a coordinate system that has all of the different regions of the brain mapped out. So we can basically locate specific brain regions for each animal by putting them in an apparatus called a stereotaxic apparatus, which allows the experimenter to locate specific number coordinates in X, Y, and Z planes on the animal's head, and then drop the cannula in that location. So during the surgery, the experimenter is going to locate a point on the skull that is called the bregma. This is basically a junction between some of the bones in the skull. Remember, your skull is made up of bones that have fused together. So you're going to be able to see those sutures. When the bones fuse, as I've said, they do form sutures that basically kind of look like seams on the skull. So in particular, we're going to be looking at the bregma, which is the junction point of both the sagittal and the coronal skull sutures. So here you can actually see our target of the lesion. And then just above that, here's our coronal suture. Here is our sagittal suture. This will be the bregma, and we'll drill the holes right there. So this is obviously not just going to be used for animals, but it can be used in human surgeries. Neurosurgeons will actually use a combination of stereotaxic coordinates and MRI scans for a patient before they remove a tumor or other types of tissues. In addition to lesion surgeries, we might use stereotaxic coordinates to place stimulating devices, things like deep brain stimulation, which is a treatment for Parkinson's disease, or implants that record electrical activity. So this is often going to be done in animals when we want to record neural activity once they are awake and behaving, like that monkey that we saw moving the robotic arm. We might also potentially use this for patients that have epilepsy. Now, after an ablation surgery has been performed, it's important to verify the location of the lesion when you report the results. You want to make sure that you weren't off the mark, given that every skull and every brain is going to be different on that from the stereotaxic map. And so to do this, scientists will do what is called fixing the brain. They're going to replace the blood with chemicals like formaldehyde that preserve the brain tissue and actually allow the brain to be handled, and it won't fall apart when you cut through it. So it's very similar to what you would see if you were dissecting an animal. They are often fixed with formaldehyde. The fixative process is going to harden the brain tissue, and it's going to prevent it from decomposing. So once you've done that, you're going to take slices of the brain according to stereotaxic coordinates. So you have all of these little thin slices with labels for where they're located in the brain. Then you're going to stain them. Now, depending on certain cases, you may want to stain the cell bodies. Where there is damage in the tissue, you're going to see a hole in that stain. And then finally, you're going to view these under a microscope to determine the exact location of the lesion as well as its size. And then you would basically throw out or basically not include the results of any animals whose lesions were too far off from the rest of the group. Okay, so that's pretty much it for stereotaxic surgery, but we still have more to talk about. So just as a reminder of what we are talking about, we've talked about ablation, we've talked about EEG and MEG. Now we're going to largely focus on neuroimaging. So we're going to talk about indirect methods of neuroimaging, including fMRI and PET scans. And then we'll also look at structural neuroimaging, where we're actually looking at the volume and the structure of brain areas. And we'll talk about CAT scans as well as MRI. So what is neuroimaging? You've already heard me talk about this. Neuroimaging is basically, you can divide it into two major branches, as you've already kind of heard me talk about. So we have structural neuroimaging and we have functional neuroimaging. So structural neuroimaging occurs when we are looking at the body's anatomy. So we're going to be looking for things like tumors, aneurysms, strokes, other types of brain damage. And basically the two major methods that we use to assess the structure includes computed tomography or CT or CAT scans and magnetic resonance imaging or MRI. And then we have functional neuroimaging. So we've talked about structure. Now we need to talk about function. Function is all about the body's activities. So this could include things like metabolism, blood flow, binding to receptors. And the two major methods that we're going to use for this are functional magnetic resonance imaging or fMRI, as well as positron emission tomography or PET or PET scans. So we're going to start by talking about a CAT scan. So CT scans are sometimes called CAT scans. They've been around since about the 1970s. And in general, if you have a broken arm or if you have a tumor, odds are if your facility doesn't have an MRI, it is going to have a CAT scan. So basically you are going to lie on a table inside a donut-shaped machine. It's going to look like an MRI scanner, but it's a little different. An X-ray tube inside of the machine is going to rotate around your body and send small doses of radiation as it passes through various angles. So as those X-rays pass through your body, different tissues absorb different amounts of radiation. And detectors inside of the CAT scanner are going to measure the radiation that has passed through your body. And it's basically going to convert that absorption rate into an image that shows the different tissues of the body, including the brain. So basically, CAT scans are going to build an anatomical picture of an area of the body under investigation. So generally, the job of CAT scans are to look at different abnormalities. So here is an example of a CAT scan. Now, some of you may be wondering why this is different from an MRI scan. And ultimately, here's how you know it's a CAT scan. CAT scans can only look at the brain like this. MRIs are capable of looking at the brain in X, Y, and Z coordinate planes, but this is the only cut you're going to see. This is the only plane that you can view a brain from if you're looking at CAT scans. So you don't get all 3 dimensions here. So here's kind of another example of how it works. So you're laying inside the scanner. The X-rays are going to pass around your body and different tissues are going to reabsorb at different rates. So what you can kind of see here is that the skull is very, very intense. That's very white. The ventricles are very dark. And then different areas of the gray and white matter are different shades of gray. Now, if you look very carefully, you're going to notice there's a hyperintensity right here. That means that there's something pretty solid there, and it probably means that there's a tumor in one of the ventricles. So really quick, you are looking at what are called Hounds field units. This is basically a measure of how much passes through or how little radiation is absorbed. And what you're going to see is that in general, air is not very dense. And so kind of going back really quick, you can kind of see right here, those are the sinuses of the nose, and you can see that there's a lot of air there. As a result, it's very dark. Nothing is absorbed there. So because of that, air is going to show up very, very dark. And then we have things like fat, and then water. Here's gray matter and white matter, and then blood, bone, and especially dense bone. So basically, the more solid something is in a CAT scan, the more intense it is going to appear. So let's talk about what this all means when we're looking at these scans. So bone, brain, and blood absorb different amounts of radiation. Bone absorbs the most. It's going to be the brightest and very little radiation passes through. Air and cerebrospinal fluid absorb the least, so those are going to be the darkest. And this actually allows us to identify abnormally located tissues or blood. So here in panel 5, you can actually see a white spot in the parietal lobe. Now, it looks left, but because of the way that this is shot, that is actually the right side of the brain. This is the standard orientation for scans. This in particular is due to a stroke, the kind where we would actually get bleeding in the brain. Blood is going to be more dense to the X-rays that are passing through, so it appears brighter because it absorbs more. So CAT scans, up until very recently, were the most common type of scan administered to patients to investigate neurological problems, but they've become less popular because of radiation and because, as we're about to see when we talk about MRI, the resolution's just not as good. They can only be taken in one plane. You are only viewing horizontal slices of the brain. So that's how you know it's a CAT scan. If all you get are horizontal slices of the brain, like you see here, and that's it, you're probably looking at a CAT scan. So It's more informative to see all planes of view, and that's why MRI has become as popular as it is, because it does allow you to do that. So finally, I want to finish up. So what you're looking at is an image of somebody who actually had a bullet go through their brain. And you can see here, you can actually follow the track of the bullet. The holes are basically going to fill with cerebrospinal fluid, which you see right here. So this is pretty much all that I had. We will talk about the physics behind MRI and fMRI in class tomorrow, and I can't wait to see you then. Bye.